Protein structure and folding is a lecture 3 in session 2 of Molecular Gene and Disease module. This lecture presented by Dr. Zainab Al-Manasi. The learning objectives is to describe what is meant by the primary, secondary, tertiary, and quaternary structure of the protein. Describe the types of bonds and forces that involved in protein structure. And the last learning objective is try to explain the key features of the two major secondary structure elements of proteins, alpha helix and beta sheet. Peptide and proteins. In order to understand proteins, first of all, we need to define the peptide bond. Peptide bond is a chemical bond that formed between two molecules. The carboxyl group of one molecule, this reacts with the amino group of the second molecule, releasing of water, H2O. So, this is the first amino acid. The carboxyl group of this amino acid will react with the amino group of the second amino acid. The result of this reaction forming a peptide bond between these two amino acids and releasing of water. The amino acids in peptide chain is named as amino acid residue, and each peptide chain may consist of 2 to 50 amino acids. These amino acids are linked by peptide bonds and to form a polypeptide chain, and then these chains can twist to form the 3D protein shape. So these are different amino acids which bind with each other to form a peptide or a polypeptide, and this polypeptide is twisted to form a protein. Therefore, polypeptides have two terminal ends. One end is ended by the amino group, and the other end is ended by the carboxyl group. In addition to that, the sequences of the amino acid is written from left to right. Regarding proteins, proteins are organic polymers composed of amino acids ranged between 50 to 2000 amino acid residues. The mean molecular mass of an amino acid residue is about 110 Dalton units. So therefore, the molecular mass of protein between 5.5 and 220 kilodalton. So this is a primary structure of the protein, which composed from amino acid that linked to each other by a peptide bond. This primary structure, when it's folded, forming a tertiary structure of a protein. So this is a protein. Therefore, protein do not exist as a linear polypeptide, but instead it folds into a unique three-dimensional structure. There are two general classes of protein molecules. The first one is a globular protein, and the second one is a fibrous protein. The characteristic features of a globular protein is compact, soluble, and spherical in shape. Best example, myoglobin. While fibrous proteins are typically elongated and insoluble in water. Best example is collagen. Therefore, globular and fibrous proteins may exhibit in one or more of four types of a protein structure. This slide showing the difference between globular and fibrous protein. So as you see here that the fibrous protein is different from the globular protein in shape. Fibrous is long and narrow, while globular is round and spherical. The purpose of the fibrous is structural, while globular is functional. The acid sequence in the fibrous is repetitive amino acid sequence, which means that regular, while in the globular is irregular amino acid sequences. And this irregularity due to because of the amino acid present, they are either polar or non-polar amino acid. So the irregularity in the sequences of these amino acids will create a force. That force will lead to the folding of this structure of the protein to form round or spherical structure. The durability of the fibrous protein is less sensitive to changes in the pH, temperature, and etc., while globular protein is more sensitive to the changes in the pH and the temperature. The example of fibrous protein: collagen, myosin, fibrin, actin, keratin, elastin, while globular like enzymes, hemoglobin, myoglobin, immunoglobulin, and insulin. The solubility: the fibrous generally insoluble in water, while but globular generally is soluble in water. So the shape of the protein is very important for defining or for determining the protein function. So as you know that the linear sequence of the primary protein is composed from linked amino acids. These amino acids contain the information that are necessary to generate a protein molecule with a unique three-dimensional shape. And this unique three-dimensional shape is responsible for determining the function of the protein. We have four types of a protein structure. The first one is the primary, which is a linear polypeptide chain, and the bonds between these amino acids is a peptide bond. It is a covalent bond. This converts into secondary, which is a twisted form of the protein, and this hold together, the bond between, responsible for this twist form of secondary protein or secondary structure is the hydrogen bonds. This will change into tertiary, which is a folded type of the protein, and then this will be changed into quaternary structure, and this quaternary structure composed from 4 subunits of tertiary protein that hold together by the by the same bonds that hold in the tertiary structure of a protein. Therefore, primary structure is a linear sequence of amino acids, and this linear sequence is referred to us as a polypeptide chain. The amino acids in the primary structure are held together by a covalent bond, which means that the peptide bond. Therefore, the primary structure of a protein starts from the amino terminal end and ends with a carboxyl terminal C end. The sequences of an amino acid in the primary structure of a protein is arranged or is unique, and this is important to define the structure and the function of the protein, because if there is abnormal arrangement or abnormal amino acid sequence, this will lead to the incorrect folding of the protein, and this will lead to the change in the structure of the protein, and this abnormal structure in the protein lead to the loss of the normal function of this protein, and at the end lead to the different genetic disease. Therefore, the importance of the primary structure of a proteins, it's very important in order to understand the genetic diseases that results due to protein with abnormal amino acid sequences. And the best example for abnormal amino acid sequences is sickle cell anemia. So, in normal hemoglobin, the DNA, it will be transcribed into normal messenger RNA, and this normal messenger RNA, it will be translated into a normal hemoglobin that containing normal sequences of amino acid. While in the mutant type of hemoglobin in the DNA, mutant type of hemoglobin DNA, this will transcribe into abnormal messenger RNA or mutant type of the messenger RNA, and this messenger RNA will translate it into abnormal hemoglobin that containing abnormal sequences of amino acid. See here, this is abnormal hemoglobin containing a valine amino acid in a state of glutamine. Therefore, the bonds responsible for the stabilization of a primary structure is the peptide bonds. And these peptide bonds are strong. They are not broken by conditions like heat or denatures agents like in the protein, like heat or high concentration of urea. But in case of prolonged exposure of these peptide bonds to strong acid or base at elevated temperature, this will lead to the hydrolyze these bonds non-enzymatically, while enzymatic hydrolysis of the peptide is done by the protease or proteolytic enzyme. So, these peptide bonds generally it doesn't affected by temperature or urea, high concentration of the urea. But if these peptide bonds exposed to the strong acid or base at the elevated temperature, it will be hydrolyzed, and the type or this type of hydrolysis is non-enzymatically. At the same time, these bonds, the peptide bond, can be hydrolyzed enzymatically by the using of the protease or proteolytic enzyme. In secondary structure of proteins, the polypeptide here forms regular arrangements of amino acids that are located near to each other in the linear sequence. So therefore, there will be two-dimensional structure. This structure is formed due to hydrogen binding between the hydrogen of the amino groups and the oxygen of the carbonyl groups. So you see here, this is an amino acid, and this is the peptide bond. This is a peptide bond between the carboxyl group of the first amino acid and the amino group of the second amino acid. And this is another polypeptide, and this is the carboxylic, or this is the peptide bond, and here this is the amino group of this peptide group. So the secondary structure is resolved by the hydrogen binding between the amino group of the first peptide bond with the oxygen of the carbonyl group of the second peptide bond. Therefore, secondary structure was a result of hydrogen bonds between the peptide groups. And therefore, we have, there are two types of structure, of secondary structure, alpha helix and beta sheet. So what is alpha helix? Alpha helix is a coiled structure that's stabilized by intra-chain hydrogen bonds. So the hydrogen bonds, it extends up the spiral from the carbonyl oxygen of one peptide bond, this is peptide bond, with the amino group, with the hydrogen of the amino group of the second or other peptide bond. So this is the structure. This is the primary structure of the primary structure of polypeptide. So when there is a reaction between the oxygen of the carbonyl group of one peptide with the hydrogen of the amino group of the other peptide bonds, this reaction or this link is called hydrogen bonds, forming a secondary structure which is called alpha helix. The characteristic features of alpha helix that there is the amino acid spaced 3 or 4 apart in the primary sequence. You see that this is the primary sequence. So every 3 amino acids, the hydrogen bond will form. So thus amino acids spaced 3 or 4 apart in the primary sequence are close together in the alpha helix. So these amino acids which are apart in the primary structure, they will be close to each other in the alpha helix. It is the most common polypeptide helices, and it is a stable, rigid conformation. Amino acids that disrupt an alpha helix, for example proline, it cannot donate an amide hydrogen bonds. So this is first peptide bond. This is the first, this is the peptide bond, the carbonyl group of peptide bond. It will react with the oxygen of this carbonyl group. It will react with the hydrogen of the amino group in the other peptide bond. In case of a proline amino acid, it cannot donate this hydrogen in order to forming a hydrogen bond with the oxygen of the carbonyl group of the other peptide bond. The other amino acid is a tryptophan, and this is because of steric interference. And what I mean by steric interference are non-bonding interactions that influence the shape. Other types of amino acid are branched amino acid, for example valine and azoleucine, because also of their steric interference, and acidic and basic amino acid because they form ionic bonds or electrically ionic bonds and electrically repel each others. Regarding the data sheet, all of the peptide bond components are involved in hydrogen bonds. And if you see here that the amino acid residue of the beta sheet form a zigzag shape, and this is because in which here that the R group of adjacent residues is in opposite direction. So you see here that this is the first amino acid. You see that the R group, the side chain of this amino acid, it's in opposite direction of the other R group of other amino acids. So this is the difference in the position or in the direction of the R group will lead to the formation of the zigzag. Therefore, beta sheet may be parallel or anti-parallel. Parallel, which means that they are run on the same direction. Anti-parallel, they are run in opposite direction. Parallel beta sheet, here the adjacent beta strands run in the same direction, as you see here in this figure. This is a polypeptide and this is another polypeptide that are run on the same direction. And the hydrogen bonds connect each amino acid on one strand with the two different amino acids on the adjacent strand. So this amino acid on the first strand, the hydrogen bonds will form with two amino acids in the another strands. While in antiparallel beta sheet, the adjacent beta strands run in opposite directions, and the hydrogen bonds between the amine and the carbonyl group connect each amino acid to a single amino acid in the adjacent strand. Yani each amino group and the carbonyl group of amino acid in any strand, it will be connect to the another amino acid, to a single amino acid in the adjacent strand. This is lead to the stabilizing of the structure, which means give more stabilization to the structure. Most of the beta sheets are not flat, but they have a right-handed twist. Clusters of twisted strands of beta sheet, this form the core of many globular protein. Tertiary structure of a protein, this is a folding up of the secondary structure. So here that the amino acids, which they are apart in the primary structure or in the primary sequence, may interact with each other and forming the three-dimensional structure. Polypeptide chains that are greater than 200 amino acids in length generally consist 2 or more domers. So what is domains? Domains are regions of the polypeptide that have distinct structure and serve particular rules. So for the example of the domains, for ligand binding or interaction with other proteins and extra. Tertiary structure is a result of a side chain reaction interactions. There are many bonds responsible for the formation or for the folding of secondary protein into tertiary structure of a protein, but the main bonds here, or the main interaction, is a side chain interaction. Tertiary structure of a protein is stabilized by different types of bonds and interactions between the amino acid R groups. So these include hydrogen bonds, covalent disulfide bonds, hydrophobic interactions that originated from the R groups, side amino acid R groups, and ionic interactions in addition to van der Waals interaction. So for example here the myoglobin, it's one polypeptide chain and it is a monomeric protein. This is the tertiary structure of the protein, and you see here that the different types of the interactions and bonds that are responsible for the stabilization and forming the tertiary structure of the protein. And these bonds are, the first of the bond is a peptide bond that originated between the amino group and the amino acid, The second bond is a disulfide bridge. The third one is the hydrogen bonds. And the fourth one is the ionic bonds. And in addition, this is the important bond that's responsible for the folding of the amino of the protein and of the polypeptide chains, which is a hydrophobic interaction, and this is due to the presence of the R group, the amino acid R groups. So what is protein folding? Protein folding is the process by which protein structure assumes its functional shape or conformation. This means that by folding of the protein or by folding of the polypeptide into a specific three-dimensional shape, this protein will perform their biological activity or their biological function. This means that this type of a protein, after folding, become functional or become active. Then, unfolded or misfolded of this protein will contribute to different pathology of many diseases. Therefore, and protein folding occurs in a cellular compartment called endoplasmic reticulum. The classical principle of a protein folding is that all the information that is required for a protein to adopt the correct three-dimensional conformation and to be a functional and active protein is provided by its amino acid sequences. So for that reason, the arrangement or the sequences of the amino acid is very important in determining the shape and the function of the protein. Chaperones here are proteins that facilitate the folding of other proteins. How the protein is folded? You see here that the side chains of highly polar amino acid, it tends to be in on the exterior of the proteins and form a bond with the hydrogen bond with the water, while the side chain, which are non-polar for the non-polar amino acid, are normally clustered. They are hydrophobic and they are normally clustered in the interior side of the protein to shield them from the water. So there are different types of the amino acids. Some of them they are hydrophobic and some of them hydrophilic. So those for hydrophilic, which means that like water, they found on the exterior side of the protein in order to react with the water, while those side chains of the amino acid which are hydrophobic to water, they are present normally clustered or present in the interior of the protein and in order to shield them from the water. So for that reason, this clustering of these non-polar amino acid side chains lead to the formation of the tertiary, or lead to the folding of this protein and forming the tertiary structure. The quaternary structure is involves the association of two or more polypeptides into functional proteins. So these separate polypeptides are held together by the same intermolecular forces found in the secondary and tertiary structure. And the best example here for the quaternary is the hemoglobin protein. So you see that quaternary structure is composed from tertiary protein, and each one of these called or named is subunit, and this subunit is a tertiary protein, which means that the quaternary is formed from the 4 subunits of tertiary protein. In a quaternary structure, the polypeptide chains may be identical. In this case, we named or is known as homomeric, or could be different, it's heteromeric. Some of the proteins contain covalent disulfide bonds, and these bonds link the sulfhydryl groups of the cysteine residue. The intrapolypeptide disulfide bonds that stabilize the 3D structure of the peptide, while the interpolypeptide disulfide bond that between the subunits, it will stabilize the quaternary structure of the proteins. What is protein denaturation? Protein denaturation is the loss of a protein structure sufficient to cause the loss of function. This is a process called denaturation. So denaturation is brought by breaking the bonds that hold and maintain the protein's tertiary and secondary structure. So the bonds of the protein, peptide, ionic, disulfide, hydrogen, and hydrophobic. So any factor that lead to hydrolyze or break down these bonds, breaking these bonds will lead this called denaturation. Denaturing agents include heat, organic solvents, mechanical mixing, strong acids or bases, detergents, and ions of heavy metals such as lead and mercury. And in this example, here is a tertiary protein. This tertiary protein is treated with the beta-mercaptoethanol, which is a reducing agent. This beta-mercaptoethanol lead to hydrolyzing the disulfide bond here and changing that tertiary protein into primary or inactive or denatured types of a protein. So beta-mercaptoethanol will reduce the hydrolyze or will lead to the hydrolyzing of the disulfide bond and of this tertiary protein, and then this protein become unfolding or misfolded, and then it become biologically, that means that it will change the structure of this protein, and then this type of this because of the changing in the structure of the protein or in the shape of the protein, there will be this protein become inactive or biologically inactive. Thank you very much for listening, and if you have any question or if there is any confusion, please don't hesitate to ask. This lecture about hemoglobin and myoglobin, which is lecture 4 in session 2 in molecular gene and disease module, presented by Dr. Zainab Al-Manasir. The learning objective of this lecture is a continue to the learning objective of lecture 3. So in this lecture we try to explain the physiological rules of myoglobin and hemoglobin, contrast the oxygen binding properties of myoglobin and hemoglobin, and explain why hemoglobin is most suited to its role as an oxygen transporter. Also describe the major structural differences between oxygenated and deoxygenated hemoglobin, and the molecular basis of cooperativity. Learning objective number 7 is to describe the effects of carbon dioxide, hydrogen, 2,3-bisphosphoglycerate, and carbon monoxide on the binding of oxygen by hemoglobin, and the physiological significance of these effects. The last learning objective is about the mutations in globin genes which can give rise to diseases such as sickle cell anemia or thalassemia. Hemoglobin is an iron-containing protein that transports oxygen in human blood from the lungs to tissue of the body. So, hemoglobin means heme plus globin. Each hemoglobin molecule is made up of 4 units of heme, so this the green color units is a heme unit or heme group, iron group, surrounded by a globin protein groups. So what is heme? Heme is an iron porphyrin compound, and porphyrin is a tetrapyrrole structure. These are pyrrole structure. The ferrous iron occupies the center of the porphyrin ring and establishes 4 nitrogen bonds of all the pyrrole rings. Therefore, iron attached to 6 coordinated bonds, 4 coordinated bonds planar with the nitrogens, 1 coordinated bond linked to oxygen, and 1 coordinated bond linked to histidine amino acid of alpha or beta globin chain. The central iron provides reversible binding to oxygen and carbon dioxide molecules. So this is the structure of a heme, which consists from 4 pyrrole rings. In the center there is an iron, and this iron makes a linkage with the 4 linkage with the nitrogen, and 1 linkage with the globin, with the amino acid histidine that present in the globin, alpha and beta, and also 1 link with the oxygen, and this linkage is reversible, which means it can be bind with the oxygen and bind with the carbon dioxide. So heme groups can be present in other compounds rather than hemoglobins, like myoglobin, cytochromes, and peroxidase. Globin, hemoglobin comprises 2 pairs of globin chains that are twisted together, and in normal adult hemoglobin there are 141 amino acids in each alpha chain and 146 amino acids in each beta chain. Therefore, the complete structure of the hemoglobin is 4 heme units surrounded by 4 globin units, and each heme can carry 1 molecule of oxygen. What are the main types of hemoglobin? The first one is hemoglobin A1, and this is alpha 2 beta 2, represent about 97% of total hemoglobin, and consisting of 2 pairs of globin, which are alpha pair and about 141 amino acid residue, and beta pair 146 amino acid residue. The other types of hemoglobin is hemoglobin A2, and this is represent about 2 to 3% of the total adult hemoglobin. It consisting of 2 pairs of globin alpha 2 and delta 2. Third type of hemoglobin is hemoglobin F, which is a fetal hemoglobin. This hemoglobin consisting of alpha 2 and gamma 2 is present primarily in the embryonic life and usually disappear from the circulation by the age of 6 months. Hemoglobin F represents less than 1% of the hemoglobin in adults, and it has a greater affinity for oxygen than adult hemoglobin. The last type, or the fourth type of hemoglobin, is GAWAR1 and 2. These are synthesized by the embryonic yolk sac during the first month after conception. For hemoglobin E GAWAR1, consisting of zeta-2 and epsilon-2, while hemoglobin E GAWAR2 consisting of alpha-2 and epsilon-2. This figure showing that the three types of the hemoglobin, the embryonic hemoglobin, fetal hemoglobin, and the adult hemoglobin. This table shows the difference of the hemoglobin in the composition and in the percentage. So for the hemoglobin A1, it composed of alpha-2 and beta-2 and represent about 97% of the total hemoglobin. Hemoglobin A2 consisting of alpha-2, delta-2 and represent about 2%. Hemoglobin F consisting of alpha-2, gamma-2 and represent about 1% of the adult hemoglobin, while at birth is about 80%. And as I mentioned before, that hemoglobin F disappears from the circulation by the age of 6 months. Regarding globin synthesis, globin is synthesized by the ribosomes in the cytosol. And there are two genes located on chromosome 16 responsible for encoding of the alpha-2 chain, while the other 4 genes located on chromosome 11, they are responsible for the encoding. One gene for the beta chain and one gene for the delta chain and 2 genes for the gamma chain. So in the embryonic hemoglobin, which is GAWAR1, this consisting of zeta-2 that present on chromosome 16 and epsilon-2 present on the chromosome 11 that encoded from the gene present or located on chromosome 11. While GAWAR2, alpha-2, this is transcribed from the gene alpha gene that located on chromosome 16, and epsilon-2 transcribed from a gene located on chromosome 11. Regarding fetal hemoglobin, hemoglobin F, consisting of alpha-2 and gamma-2. Adult hemoglobin, either hemoglobin A1, this is alpha-2 beta-2, or hemoglobin A2, which is alpha-2 delta-2. Regarding heme synthesis, heme synthesized in the bone marrow and in the liver. Their site of the synthesis is in the mitochondria and partly in the cytoplasm. The substrate for heme synthesis is succinyl-CoA plus glycine, and the pathway or the process for heme synthesis consisting of 8 steps. Myoglobin is a polypeptide, is a single polypeptide, structurally similar to the hemoglobin, but the hemoglobin is tetramer, myoglobin is monomer. So because it is a single polypeptide, it can bind only to one molecule of oxygen with high affinity, and the function of the myoglobin is for storage of oxygen inside the muscle and to be used later on when the partial pressure of the oxygen be very low. This table reveals the difference between hemoglobin and myoglobin. So the number of chains in the hemoglobin, 4 polypeptide chains. In myoglobin is single polypeptide chains. Therefore, hemoglobin is a tetramer, while myoglobin is a monomer. Hemoglobin can bind to the carbon dioxide, carbon monoxide, nitroxide, oxygen, and hydrogen, while myoglobin binds to oxygen, tightly and firmly. Hemoglobin presents systemically in all over the body, while myoglobin in the muscle cells. The type of the curve of the oxygen dissociation curve for hemoglobin is a sigmoidal in shape, while in the myoglobin is hyperbolic curve. The abbreviation of hemoglobin HP and the myoglobin MB. The role of hemoglobin is to carry the oxygen, while the myoglobin is to store the oxygen. Concentration of hemoglobin in the RBC is high, while the myoglobin is low. Hemoglobin molecule can exist in two forms or in two states. The first one is the T-tense state or TOT. This is a deoxyhemoglobin, which means that lacks of the oxygen, and here the globin units are held tightly together by electrostatic bonds. The other form of the hemoglobin is the R state or relaxed, and this is in oxyhemoglobin, which means that fully oxygenated. The bonds here are broken when they are bound to oxygen, and it has a high affinity for oxygen than in T form. Cooperative binding of oxygen to hemoglobin. This features is to change the conformation of the hemoglobin or change the state of the hemoglobin from the T state to R state. So, when oxygen molecule binding to the hemoglobin, to the first heme, this will lead to altering in the conformation or alters the conformation of the hemoglobin that allow the other units to bind with oxygen, which means that change the state of the hemoglobin from the T state to R state in order to bind more oxygen. And conversely, when each molecule of oxygen released from the hemoglobin, this will change in the shape or in the conformation of the hemoglobin and make it easier to other oxygen to release from the hemoglobin, which means that conversely, each oxygen molecule is released. The change in hemoglobin makes it easier for other molecules to be unloaded. The oxygen dissociation curve, it's a curve that plots the proportion of hemoglobin in its saturated form on the vertical axis against the partial pressure of the oxygen on the horizontal axis. And as you see here that for the hemoglobin, the curve of the oxygen dissociation is sigmoidal in shape, while the curve of the myoglobin is hyperbolic in shape. There are certain factors affecting the oxygen dissociation curve of the hemoglobin. First of all, if shift the oxygen dissociation curve to the right, this means that the hemoglobin has low affinity to oxygen, and this effect we call it Bohr effect or known as Bohr effect. This effect is response to high temperature, high PCO, high partial pressure of carbon dioxide, in case of decrease in the pH, and increase in 2,3-diphosphoglycerate, and the other name is 2,3-bisphosphoglycerate. While if the curve of the oxygen dissociation curve shifting to the left, this means that the hemoglobin has a high affinity to oxygen, and this effect we call it or known as Holden effect. This effect is a response to the low temperature, low partial pressure of carbon dioxide, high pH, and low level of the 2,3-diphosphoglycerate, and in case of carbon monoxide poisoning. So what's Bohr effect? Polar effect is a decrease in the oxygen affinity of a hemoglobin, and this occurred in response to increase in the temperature, increase in the partial pressure of the carbon dioxide, and increase in the 2,3-diphosphoglycerate. Also, in case of low pH. 2,3-diphosphoglycerate, this is a special intermediate of glycolysis in erythrocyte, and this plays an important role in enhancing or in liberating of oxygen from the hemoglobin. So this is a dissociation curve, oxygen dissociation curve. This is the vertical axis represent the oxygen saturation of the hemoglobin, while the horizontal axis represents the partial pressure of the oxygen. So you see here that at 100 millimeter mercury, and that is in the lung, the oxygen saturation of the hemoglobin about 100 or less than 100, 98 to 100, and this is for the normal curve, for the normal person in resting state. While in case of exercise or in the contraction of the muscles, we see that increase in the temperature and increase in the partial pressure of the carbon dioxide, and this will lead to the increase in the hydrogen ions, and the hydrogen ions lead to the decrease in the pH, and at the same time increase in the 2,3-diphosphoglycerate. So these factors will affecting the curve and causing shifting of the curve to the right side. So this means that here the hemoglobin become less affinity to the oxygen, and it's ready to liberate of the oxygen to the tissue. In Holden effect, it's the opposite to the Power effect. Is an increase in the oxygen affinity of the hemoglobin, and this promotes the release of the carbon dioxide in response to decreased temperature, and decrease with carbon dioxide, increase with pH, decrease with 2,3-diphosphoglycerate, and in case of carbon monoxide poisoning. So again, this is an oxygen dissociation curve. The vertical axis represents the oxygen saturation of the hemoglobin, the percentage of the oxygen saturation of hemoglobin, and the horizontal axis represents the partial pressure of the oxygen. So what happened here? In case of low temperature or low carbon dioxide, and in case of high pH and low 2,3-diphosphoglycerate, and also in case of high carbon monoxide poisoning, the curve will be shifted to the left, and this means that the hemoglobin has a high affinity for oxygen, which means that the hemoglobin is not ready to release the oxygen. It will keep the oxygen، يعني يحتفظ بكمية الأكسجين الموجودة، ما يطرحها. So this is called the Holden effect. So there are two questions here. Why 2,3 is phosphoglycerate decreases the oxygen affinity of hemoglobin, and why patient with carbon monoxide poisoning has a cherry red or pink cheek appearance? The answer of the first question is because 2,3-biphosphoglycerate, bisphosphoglycerate, decrease the oxygen affinity of the hemoglobin because this compound binds with the deoxyhemoglobin but not with the oxyhemoglobin. 2,3-bisphosphoglycerate binding with the deoxyhemoglobin and not to the oxyhemoglobin. This binding will stabilize the deoxyhemoglobin in a T state, يعني keep the hemoglobin in a tense state, in a T state, so not allow the oxygen so for that reason it will be decrease the oxygen affinity. So this means that it prevents the heme site to bind with the oxygen. While in case while in carbon monoxide poisoning, the answer for the carbon monoxide poisoning, because carbon monoxide, when it's bind to one or more of the heme molecules or heme site, this will lead to the shifting of the hemoglobin from the tense state into a relaxed state. This shifting from the tense to the relaxed, causing that the remaining site of the heme to bind with the oxygen with high affinity. The other question is why hemoglobin F has high oxygen affinity than hemoglobin F has high oxygen affinity than hemoglobin A. This curve showing that the adult hemoglobin and the fetal hemoglobin. What I mean here that the oxygen dissociation curve of the fetal hemoglobin and the oxygen dissociation curve of the adult hemoglobin. The answer for this equation is that because hemoglobin F having a gamma subunit, and this gamma subunit allows to bind more strongly with the oxygen. While the second answer is because the presence of 2,3-bisphosphoglycerate. This compound interacts much more with the hemoglobin A than hemoglobin F. Why? Because the adult beta subunit has more positive charges than the fetal gamma subunit. And these positive charges attract the negative charges from the 2,3-bisphosphoglycerate. Therefore, the preference of 2,3-bisphosphoglycerate for hemoglobin A makes that the hemoglobin F binds to oxygen with high affinity. Hemoglobin abnormality, which is a variant form of hemoglobin that is often inherited and may cause a blood disorder. These disorders could be in the heme production and that lead to the porphyria, or could be in the globin synthesis, and globin synthesis either affecting the structure, for example sickle cell disease, or affecting the production of the globin like thalassemia. Porphyria is a group of disorders that caused by abnormalities in the chemical steps that lead to the heme production. So as I mentioned before that synthesis of the heme is occurred by 8 steps, part of them in the mitochondria and part of them in the cytoplasm. So any defect in each one of these 8 steps will lead to the disease or lead to the defect in the heme production. So, and this will lead to the porphyria. Porphyria, there are many types, but the most common type of porphyria are acute intermittent porphyria, and the second type is porphyria cutanea type. Regarding the abnormality in the globin synthesis, and as I said before, in the structure, which lead to the sickle cell anemia, and this caused by a mutation in the beta globin gene that change the position of the amino acid, the 6th amino acid, from the glutamic acid to valine. Glutamic acid is a charged amino acid, to valine, which is a non-polar amino acid. Normally, in a normal hemoglobin, the glutamic acid or the sequence of the glutamic acid is the 6th. It's a 6th amino acid. In sickle cell anemia, this 6th amino acid replaced by valine. So sickle cell anemia could be homozygotes, which means that produce only abnormal beta chain, and that lead to the hemoglobin S, or heterozygotes, which produce a mixture of normal and abnormal beta chains, and that makes normal hemoglobin A and hemoglobin S. The pathology of sickle cell anemia is when hemoglobin S is deoxygenated. So the molecules of hemoglobin will polymerize, forming structures or bodies called cytocrystalline structures, these known as dactoid. Dactoid will distort the red cell membrane and produce a characteristic of sickle-shaped cell. So this is a normal RBC. When this hemoglobin becomes deoxygenated, these RBCs, the hemoglobin in these RBCs polymerize, forming cytocrystalline bodies called or known as dactoid, and these dactoid lead to the damaging of the membrane of the red blood cells, forming a characteristic of sickle cell or sickle-shaped cells. While in thalassemia, there is defect in the production or in the rate of production in one or more of the globin chains, and this leads or results to the imbalance in the globin chain synthesis. So for the example of thalassemia could be alpha thalassemia, and this means that there is a defect in the synthesis of the alpha globin chain, or beta thalassemia, defect in the synthesis of the beta globin chain. In alpha thalassemia could be single gene mutation, that means mutation in a single gene, and this is clinically silent, or two gene mutations, and this leads to the microcytic anemia, or could be three gene mutations, which lead to the HBH disease. In this case, beta chains will precipitate as beta-4 tetramers, which means that HBH, and this leads to moderately severe conditions of alpha thalassemia. The last one is 4 gene mutation. The other name for it is hydrops fetalis, and this leads to the forming of the gamma-4 tetramers, which this is an HB part, and lead to the hydrops fetalis. In this case, it is incompatible with life, or the fetus here is incompatible with life. Well, in case of beta thalassemia, mutation in the beta globin gene, this leads to the generation of a stop codon in the messenger RNA. This stop codon will terminate the globin chain synthesis and lead to the variable reduction in beta globin output. So beta thalassemia could be thalassemia major, the other name is Colles anemia, and this is severe transfusion dependent, which means that the patient needs frequent transfusion of blood. Or it could be thalassemia intermediate, which means that intermediate in severity. Or the third type is thalassemia minor, and this is a symptomless carrier state. Thank you very much for your listening, and if there is any question or confusion, please don't hesitate to ask. Again, thank you very much.